Okay, guys. Welcome to this is gonna be scientific method, experimental design, and scientific method. We'll start with scientific method and go into experimental designs, graphing, and then also finish up with data tables and some stats. So let's go over the scientific method real quick. We know that we usually start with a problem. We identify a problem, or you develop a question. Scientists like to ask a lot of questions. They're great at asking questions and then trying to find those answers. Okay. So what we would do is once we identify a problem, we would go back and collect some information, get our background knowledge on that question, see if someone else has already answered the question, or see if someone has come close and see if they've attempted an experiment that would answer our question. Once we've researched this out, and we've read journal articles and read other scientists' explanation of similar questions, or anything that would deal with that topic, once we've educated ourselves pretty much on that topic or that question, we're going to form a hypothesis. And we're going to use the if-then statement for a hypothesis. Once we all have that, then we're going to come and we're going to do our design our experiment. Okay, come up with a way to design our experiment. Now, most of the time we rely on previous science work, previous scientists or researchers, and we look at their methods and we might adopt their methods. Okay, very few times do we actually come up with our own methods. Scientists always like to borrow and uh, use other people's ideas. We run that experiment and then we collect observations and data. This is always going to go into a data table, and you're also going to write down observation, any color changes, anything you see, smell, observe, and then also with the data table. Okay. Once we do that, we're looking for trends. We're looking for a trend in the data table. So to visualize it easier, we make a graph and we look for either a direct relationship or an indirect relationship. Now, for us, that's you're going to be similar to a positive slope or a negative slope. And then we come up with a conclusion. Did that data support our hypothesis? If it did, then we were right. If not, then we have to reject the hypothesis and then go back and try to reform our hypothesis to be answered again. So because of this process, science works very, very slow. Things are added on to our knowledge base very slow because we're always going through the scientific method. But once we actually get it right and we can prove that hypothesis, we know we've gone through all the steps, and we've researched out every particular problem and idea and stuff, and so therefore we can be confident about our um, research. Okay, we like to publish it. We like other people to read it, and a lot of times you have to in science you have to be willing to take critiques. You have to be willing to defend your arguments if you truly believe your arguments are correct. Okay, but one of the big thing is we have to be able to take critique. We have to have people say, I don't agree with that because of this reason. Now, most of the time we hear about science doing great things, and this is through what's called a serendipitous or a happy accident. So much of science actually just comes through a serendipitous or a happy accident. Okay, so for example, you guys started probably talked about Alexander Fleming last year, working with Staphylococcus bacteria, and he accidentally left his petri dish open, and he noticed that the bacteria had was dying, so to speak, and he found out there was another mold on there called penicillin, and then that's where we got our antibiotics. Okay, so a lot of this radioactivity, same thing, was discovered through a happy accident, or just scientists left some photography paper out right next to a vial of uranium salt that he didn't know was radioactive. And when he got back, his paper was marked up. It had marks on it. So again, just an accident happened, and he discovered radioactivity. Okay, let's go through. And remember with the video, folks, anytime that you need me to slow down, or you can just stop it and catch up and then hit play again. So that is the nice thing about doing a video. Let's go through experimental design. Lots of vocabulary here. So let's first of all talk about an experiment. This is just a controlled observation, okay? And it's going to test a hypothesis. This is the experiment we're going to do. Now, you can have multiple variables, but for us, we're only going to have one variable at a time. Okay, we're only going to change one variable at a time. All right, we have two types of variables, independent and dependent variable, or the IV and the DV. Independent variable is the one that you are going to control or you are going to change. Okay, this is the one variable that there'll be multiple levels of it. There might be 0 grams, 5 grams, 10 grams, or it might be 30 degrees, 35 degrees, 40 degrees, 45 degrees. These are the levels that you are going to test your experiment at. Okay. Now, the dependent variable is the data that you're going to measure or the data that you are going to collect. Right? You are going to collect or measure the data. You're going to make observations, and this is the data you are going to collect. Okay? So you change the independent variable, and you look to see if that change drives or affects the dependent variable. There's two groups within our experiment. There's the control group. This one has the independent variable in it. Okay? And this is testing if the independent variable affects the dependent variable. Okay? And then we also need to have a control group or a reference group. And the reference group is called the control group. And this is one that has no IV or 0 amount of your IV. Okay, so this is usually going to be water, or it's going to be no independent variable. Okay, so for example, if I was testing, does the amount of salt affect the freezing point of ice? My control would be 0 salt, and then my independent variables would be 5 grams, 10 grams, 15 grams, 20 grams. Okay, so look for 0 or water. All right, if I want to test the effect of acid on seed germination, my control would be I would use water, a neutral water solution, and see how does the seed do in just neutral water, and then I could have an acid solution. 1 molar, 2 molar, 3 molar. Molar is a unit for concentration. Now, so the only thing you can change is the independent variable. The dependent variable may or may not change. It depends how those things are related. Everything else is constants. These are not allowed to change. These are held constants. Okay. So remember, we were just wanting to see the effect between the IV and the DV, no other variables. So that's why we can only change one variable. Now, title lesson hypothesis. The title, you will always see this on the SOL, and this will really help you kind of, on those multiple choice tests, really pick out the IV and the DV. So we always write the title as the effect of the independent variable on the dependent variable. So all you have to do is input the independent variable and the dependent variable into this sentence. And it's always the effect of temperature on dissolving sugar, okay? Or the effect of salt amounts on freezing temperatures, okay? So once you write that title, you can pick out the independent variable and the dependent variable. The hypothesis, this is what we call the educated guess, all right? And we're going to use what's called the if-then statement, all right? And it goes, if the independent variable is changed a certain way, so if I increase temperature or if I decrease temperature, okay, then we're going to say then the dependent variable will change this way. And this is, again, why science kind of goes slow, moves slow in the process, is because you're testing one independent variable and one dependent variable, and you're testing the independent variable a very certain way, and you're measuring that dependent variable a very certain way, okay? So if I increase my amount of salt, then my freezing point will increase. Okay, and you would run that, and maybe you find that that doesn't work, so you got to go back and reformulate your hypothesis. If I increase my salt concentration, then my freezing point will decrease. Now, the number of the trials, the more the better. However, we don't have a lot of time in a laboratory class to actually do multiple trials. So the more the better. And this will allow you to be accurate, have a more accurate results. Okay? More accurate, or we want to be confident. We want to make sure that that first time we did it, it just wasn't a fluke. It actually we need to repeat it. Okay, so here what you're going to see is words like accountability, to validate, to be confident, to make sure. Validate is one of the ones that shows up on the SOL. We want to validate our results, so we do it multiple times. Or what we might do actually in the chemistry classroom in lab is we'll have the 6 lab tables do the same experiment, and then we'll see what everyone gets. And if we all get the same answer or similar, very close, then we know we can validate that. Conclusion, this is just the judgment based on the data obtained. Okay, remember the hypotheses are never really proven because they are always subject to additional research. And very important here, any data is always useful. Even if you say, you know what, there's no effect between salt concentrations and freezing point. Okay, that does tell us something. There's no relationship, so we don't have to worry about it. So in 0 or no results, that is data. Now, theory and scientific law. So once we've done this hypothesis and we've done it and done it and done it, tested it and tested it, and we've written a paper on it, and other people have reviewed that paper, and then other people